If not, it can really be restricted to like even just the top layer. But we quantified the visibility of the water, so to say. And when we didn't see the dolphins the moment they went underwater, because they usually stay at a depth of 1 meter almost the entire time. If we couldn't observe them at that depth, we didn't include the data in our study. But so it depends a lot. I think on average about 2-3 meters, but could go up to 7 meters.
Okay. Do you think this can affect your estimates, your behavior estimates? Because there might be, I mean, diving deep might be associated to feeding on demersal fish.
I really agree. Our study is also limited to coastal areas.
Yeah.
So
it was my
all
next
of our
question.
data, but it's a good remark because all of our data is restricted to coastal areas. Mm-hmm. We only observed them 400 meters, but on some occasions the dolphins went deeper into the strait and we were able to follow them to about 2 kilometers away from the coast. And so we saw that when they go further away from the coast, they actually start diving. So the species is said to be a coastal species, but we're not entirely sure if that's because when they move away from the coast, they start diving and they're observed a lot less than when they're at the coast.
Yeah.
But it is a limitation of the use of drones.
Yeah, that's something that is also very, very interesting because I was wondering maybe if you don't overestimate the association to kelps, because kelps by definition occur on the coast, and you also occur on the coast with your car. You're more likely to observe them associated to kelps because that's where you are.
Yeah. That was an assumption. Well, we decided to go with a coastal design because all previous literature
makes perfect sense. I don't blame you for that. But how could you? Do you have any idea how you could estimate this bias?
Because scalps are coastal.
Yeah.
But it's hard. I mean, you could work with acoustics, for example, to see how far they are away from coast to acoustics. And I know there's some studies starting to work on acoustics of Peel's dolphin. One study came out, I think, only in March this year. So it's really a new topic that started to actually address how they vocalize. And there is a new study going on next year on new study on the effects of Peel's dolphin and acoustics in the north that we will maybe be a part of. But yeah, I think acoustics is.
It's all right. Yeah, it's what you did is, I think it's super interesting, but you always wonder if there's not a bias if the night behavior is different. Yeah. They use acoustic for locating the prey or other visual predators.
Yeah, echolocation.
So they basically don't care if it's daylight or not.
No, that's true. And all behavioral budgets, well, until now, it's usually described as a daytime behavioral budget because we have no idea what happens during the night, and you would have to make like a correlation between the vocalizations and the behavior using drones and acoustic devices at the same time.
Did you hear of any accelerometer studies?
Sorry?
I mean, I've seen that for pinnipeds, for seals.
Yeah.
And they can equip the seals with accelerometer.
Yeah.
That will measure the acceleration, basically. And you can infer from the acceleration in 3 dimensions the behavior. So I don't know if that has already been done on dolphins.
I think it's
probably not on your species.
No,
definitely not on your species, but maybe on other species. And
seeing how they behave in the water, I have no idea how it will work to actually place a device on them. Yeah. Because at one point we also saw a dolphin that had, for example, plastic around its neck, and we were looking at maybe and how to remove it, but no one had any experience with catching dolphins, and everyone was like, we don't know
some people
how that would work. Well, maybe, but in Patagonia it was, it was limited.
Yeah, I think biologging approaches can be interesting. Thank you very much, man.
Thank you.
Yes, okay. Thank you for the presentation. It was very clear and enjoyed it. I have more curiosities than a question. The first one is regarding when did you were recording the data? At what season of the year? Because it was a little bit particular that when you compare your data of sexual behavior with the ones of preceding years, they have no record of that. So could it be not because they are different years or different locations, but because you were recording at a time when they have more sexual activity, which is an important part of that? So can you have any kind of bias, or have you been getting a wide season? And just to add to that, for instance, in the same table, it says that feeding and travel behavior. they are very homogeneous, so it will be independent of any kind of functionality.
Yes.
So this is my first question. The second one, yes, both of them are together.
So our data was collected in the austral summer, between January and April. And well, we recorded sexual behavior, which is something that, well, Sarah, who worked with me, has done her thesis on the sexual behavior. But it's more that sexual behavior in previous studies has been something that has rarely been described because it's extremely hard to observe. And when it's been described, it has always been included into social behaviors. So we were able to make the distinction between the two based on the drone images.
Yeah.
And well, Sarah talked and investigated about the seasonality of sexual behavior. And there she found that it is related to this time of year, but it's something that a previous study also suggested, that there is, of course, seasonality, but well, to summarize, most studies actually are conducted in the austral summer in Patagonia.
In all the other studies.
Yeah, so
So you can guess that if you don't do the study of the recording in summer, but in winter.
it would differ, of course, yeah. And it's something that we would really like to do in the future, to make a year-round recording of behavioral data, because of course our ethogram is right now also limited to behaviors of this population and of this time of year. As we don't know, like when the subadults or the calves are born, like how the population dynamic and population itself evolves, different behaviors might occur. So of course there's a lot more work to be done in the year-round behavior.
Okay. So the second curiosity is about, so just a technical issue. So you use the drones, and how do they interfere with the dolphins? I mean,
Yeah.
can they feel that they are being prosecuted? They can be tracked. You have checked whether there is some bias by interference with the measurement system
Yeah.
and the targets.
Yeah, it's a good question. We, well, I didn't put it in the PowerPoint, but I put in the manuscript that it's really important with small cetaceans. The recommendation is to stay above 30 meters at all time. So different studies have been done. It's quite recently, but between 2020 and 2022, there's a lot of studies, mostly around Australia and New Zealand, that looked at the effect of drones on small cetaceans. And they usually found that if you stay above 30 meters, it's good. The behavior is usually not disturbed. There have been very little behavioral changes, and that's what we saw also. On a couple of occasions, we went a bit lower to actually get good footage for outreach and social activities. And then we saw a different behavioral reaction. The dolphins didn't want to surface anymore, and they scattered, and so we saw that it was quite intrusive.
So you can secure your data, the regular behavior, and not the behavior of escaping for the predator coming.
Yeah, yeah.
Okay, so thank you. That's all. My pleasure.
Thank you.
Okay, a couple of questions from me. First of all, okay, very nice work. I enjoyed it very much. Actually, I have to say that I am a marine phycologist. I never realized that there were the species of the cetaceans that live in such a strict association with kelps. But maybe, okay, in this case, what is that you mean exactly for kelp forest? I'm asking this because in the nice video that you showed, okay, actually there are kelps, but they look
of course
sparse.
We well, we made the distinction. I put it somewhere here. So we defined kelps. Sorry, it's here. In 4 different categories. So we have out, near, kelp, and fetch.
Okay.
So in the Strait of Magellan, usually the kelps, it's this big dense band of kelps that is parallel to the coastline for kilometers long. And that is what we call like really the kelp beds. So that's where we define out is if the dolphins were more than 15 meters away from that. Near is if they were between 15 and 5 meters away. And kelp is when they are 5 meters less or inside the kelps. And then patch is like I showed on the video. It's like here when there's some small spread out patches of kelp.
You're right. I forgot about the previous slide. Okay, do you also see the dolphins getting really into the canopy of the kelp, of the real forest?
Yeah, it's really interesting because in the kelp beds, they don't go so much in the kelp beds. They travel parallel and they scan sort of opportunistic for prey, like octopus or something that is on the edge. But in other areas where we went to explore around in Tierra del Fuego or in different bays, different fjords, we saw that the kelp is a lot more like this and a lot less dense. And then we see that the dolphins spend almost all the time inside the kelps, and they're really going in between.
Okay.
just like this the entire
So I
time.
wonder, so is there any chance they will also look for the crabs or
Yeah.
something that lives
Yeah. Probably.
there? Okay. So very interesting.
Yes.
Yeah, one thing, please.
Are the dolphins hunted by any predators? Like, could they use the kelp as protection from?
Yeah, that's an idea that a previous study has addressed, but there have been no official records of these dolphins having any predator. There's no sharks in the Magellan Strait, and killer whales visit the Magellan Strait, but they stay in the center. They usually travel through when they come from Antarctica to Argentine or something. But our supervisor has on one occasion recorded a lot of killer whales attacking a group of dolphins, but there's just one event in the 40 years that he's worked in the area, and no dolphins were actually killed or eaten in the occurrence. So to answer your question, I don't think they have any predators.
Yeah. Okay. Thank you.
Okay, the Valentin.
Yeah, thanks for the presentation. Yeah, well, I had a question as well about like how the drone might interfere with their behavior, but you already answered. So another question is more on how can you be sure that you are always like your footage are homogeneous? Because I guess when the dolphins start to scatter, you have to go higher, and when they gather, you have to go lower. So like, does that affect the way the expert can assess their behavior, and what can you imagine to have like a more homogenized way of having the footage?
Yeah, I agree. I agree that it is a problem when you zoom out, the behavior becomes less clear. And like luckily the drones have really good cameras, so it's possible to zoom in and to still see very well, because we went to a maximum of 130, which is the maximum allowed height to fly drones in Chile. So usually up until 130 it was pretty clear, the things we saw, especially for behavioral states or the position to kelp. But for example, for very subtle events, subtle things that happen between moms and calves or these sort of things, I think it is true that you would be less likely to detect these events. So one thing to do is to pick an individual and follow the individual and stay at a certain height and stay with that certain individual. Either you stay high all the time if you really want homogeneous effects of what happens, but then I feel like you miss out a lot. It's also difficult because these dolphins have a high fission fusion dynamic, so it's really often that groups merge and splice, and it happened about every 12 minutes that a new group joined or the group split into two. So it's really tricky to stay with both. We usually had 2 drones ready, so when the groups parted, we would launch a second drone and try to do it like that, but it is hard because it is one of the limitations.
And one idea do you think it would be possible to like track individual,
It is possible.
and then to see the number of interaction in time?
It is possible. It's what my colleague Sarah did to focus on sexual behavior. She tracked different individuals to see the sexual behavior of each.
Okay, we have a couple of minutes left. Does anybody want to ask another question?
Do you know how many pods were involved, or was it like a similar, the same pod you kept?
There's no studies whatsoever on population size of Peale's dolphins. There's one study that was done in 2016, I think, to assess their like EUCN status. Then they did it, but mostly for the Atlantic Ocean. They didn't look so much at the Magellan Strait or the fjords. During our study, we also took photo ID. So we started to build a photo ID catalog to try and answer that question. And we had some recaptures, but it's a process that's still going. Right now, there are students that are processing the images because we didn't have time to process it. So hopefully in the future we will have, but we saw in total about 250 pods, but it's very likely that the real population is a lot smaller than that we had a lot of recaptures.
Okay, then another minute.
No, it's too
Okay,
Paul Heller. Okay, so here we are implementing, so assessing post-2020 light pollution trends along marine turtle nesting beaches in Western Reunion. Okay, Clementine, the floor is yours.
Hi everyone, my name is Clementine and I'm here today to present you my master thesis, as Fabio said, about the trends of post-2020 light pollution trends along marine turtle nesting beaches in Western Reunion Islands. So let me start with a short introduction. First, we can say that light pollution is a degradation of the photic habitats by artificial illumination, and there is an important development of the coastal areas. So, and we know that light pollution is there the primary environmental stressor. Light pollution has emerged as major concern for human health, but also for its important energy consumption, the preservation of the dark sky and its effect on ecosystem and biodiversity functionality. Regarding this last point, is described as one of the most pervasive forms of environmental alteration and is doing an important evolutionary pressure on it by disrupting, for example, biological rhythm, but also daily activity and reproduction, as we will see after for sea turtles. It affects a wide array of species, including fauna and flora. Regarding its impact on sea turtles, we know that first, in the case of natural lighting conditions, female will tend to nest on dark and calm beaches. Then, when they try to reach back the ocean, they will orient toward the brightest horizon, which will be the one with the reflection of the stars and the moon on the sea surface. And also for the hatchling, when they will emerge from the nest, they will also orient toward this. In case of artificial lighting, it will disturb the orientation of the females, but first the nesting behaviors of the females, because they will abandon potentially the nesting processes, or also change the nesting site, or even throw the eggs into the water if they don't find the right place. After, when they try to orient toward the ocean, both females and hatchling would be disorientated, so they can that increase the risk of dehydration and of predation. So regarding the most problematic characteristic of the lightings for sea turtles, we know that they are particularly sensitive to cool color spectrum, so with a short wavelength lights, and also they are sensitive to high illuminance levels. So we know that in case of full moon, we'll have a maximum of 0.2 lux, which will be found. So potentially illuminances that are above that could be problematic. And the presence of multiple sources and the absence of visual obstacles will create the presence of an illuminated stretch, which will be also problematic because it will increase the visibility from the sea and from the beach. Regarding the situation in the Reunion Island, which is an island located in the Indian Ocean, we know that before the colonization, it was a significant nesting site. After, due to the intense harvesting of turtles and the nest associated with the introduction of predators, we observed a decrease in the presence of turtles in the beaches. And then during the 20th century, due to coastal urbanization, we had a near extinction of the turtles there. So what's the situation today? Due to habitat degradation, which includes the increase in coastal urbanization, coastal erosion, but also the depletion of indigenous fauna and the presence of artificial lighting and noise on beaches, we have limited nesting that occur in the beach of Reunion Islands, with about one nest per year since 2004, which is very low. So regarding this context, the Marine Turtle Discovery and Study Center decided to start research and conservation and awareness programs on sea turtles and their habitats since 1997. and they intensified the beach rehabilitation action since 2017. So they do multiple projects, but it includes plantation of endemic and indigenous species that will create natural screen against light pollution on the beach, and also different action to reduce these lights. So in 2020, they supervised first internship to assess light pollution along the western coast, and they created a monitoring protocol and also a awareness campaign about this problematic on the islands. And we know that since the end of the COVID, we have more and more tourist activities on the island, which justify the need of reassess this level in the beaches. So the different objective of our study was first to quantify light pollution level through the study of two parameters, which were illuminance levels and illuminated length. Also to look at the evolution post-2020 and to correlate it with the nesting records, to qualify the lighting parameters and their evolution, and finally to formulate some tailored lighting management recommendations. So regarding the site selection, so we based our site selection on the previous study that had been done that include all the potential or confirmed nesting sites which present some accessibility from the sea, sandy substrates, limited slope, and potentially some historical nesting records. So here we have a map of the study area that we were looking at, and in orange we have the different places we were studying from Saint-Paul to Grand'Anse, and it represents 23 kilometers of coastline that we divided into 27 sectors within 6 municipalities. So the municipality of Saint-Paul, Trois-Bassins, Saint-Leu, Étang-Salé, Saint-Pierre, and Petite-Île. Regarding our methodology, we always plan the fieldwork activity 2 or 3 days around the new moon, and we waited for to have an astronomic twilight to start the measurements. We took measurement at both top of the beach and bottom to cover the width of the beach, and we used the GPS to have the position of the points and also to estimate the distances, and we used the luxmeter for the illuminance measurements that we took all the time at 15 centimeters on the ground to have a total point of view. So in case of lighting, we were taking measurement, it's 30 meters, otherwise it was 200 meters. For each lighting, we were taking some different characteristics of the lighting, which were, for example, the type of lighting, the quantity, the color, also the visibility from the beach, and other that I'm not going to describe here. And the presence of visual obstacles that could be both natural or artificial. For the analysis, we standardized our data using Excel and Python. We used RStudio for the analysis and visualization, and QGIS for the mapping. So now I will pass to the results. So regarding the quantitative aspects of light pollution, we did this map that is an heat map that correlated the proximity of the different measurement points with the illuminance levels. And so we have in red the high light pollution level and yellow the low ones. So we can see first that the municipalities that have the higher light pollution are Saint-Paul, Saint-Leu, and Saint-Pierre, and also that the Saint-Paul municipality represents a significant portion of the total coastline that we studied. Regarding the temporal variation, we see first that we have a decrease in important places, areas that have significant light pollution, with a polarization of the high light polluted areas in the municipalities of Saint-Paul and Saint-Leu, while the municipalities of Saint-Pierre had some changing in the distribution of the light pollution. Then, on the 23 kilometers that we studied, about 12 kilometers, were illuminated, which represent about half of the total studio area. For the municipality level, we correlated the illuminated length, so in percentage, with the mean of luminance intensity that we represented here. And as you can see, the level that we found in Saint-Pierre, so the blue here, send out the other municipalities, followed by the municipality of Saint-Paul. Regarding the temporal evolution, we have some variation regarding the Saint-Pierre municipality. So we have an increase in the mean luminance level, and in the opposite, we also have a decrease in the mean luminance intensity that we took in Saint-Lô. Regarding the sector level, so in FINO scale, we can say that the sectors that present the most light pollution are the Baie Saint-Paul, Marie-Cap-Champagne, Cimetière Saint-Gilles, Les Brisants, and La Saline-Troudeau in the municipality of Saint-Paul, the city center in Saint-Lô, and then Saint-Pierre Nord, Sud, Terre Sainte, and Grandvoie for the municipality of Saint-Pierre. Okay. When we correlate both nesting records with the high level of light pollution, we found that these sectors, so the three have both high level of pollution and nesting records, and they are all from Saint-Paul. Regarding the qualitative aspects, we had almost half, more than half of the lighting that were cool in color, and that's increased compared to 2020. Almost all the lighting that were directly visible from the beach, and also 60% that had no visual obstacle, which is more important than in 2020. So regarding the discussion, we can say first that the most polluted areas belong to the municipalities of Saint-Pierre, Saint-Paul, and Saint-Lô, and especially in the touristic areas. Most of the lighting were presented some destructive aspect for sea turtles. And regarding the evolution, we didn't find major fundamental changes in the distribution of the light pollution along the coastline, except for Saint-Paul and Saint-Lô, which can be explained by differences in the lighting, so it can be some new lighting installation and stuff. And also, we had some lighting that were turned off for the public lighting in Saint-Lô, so that can explain this. And also, we had some, the thing that we can also say regarding the non-fundamental changes is that we observe some slow changes in lighting practices, because to change lighting is very expensive, so that highlight the importance of raising awareness about this problematic to be sure to have good upgrades when we change the lighting. We also have an increase in the white, proportional white lighting that can be explained by the fact that when there is some renovation lighting, they change using white LEDs because it's very economic, but we know that it's very destructive for biodiversity. So it could be more interesting to use amber LEDs. And for the lighting management, it would be interesting to prioritize the sectors that have both high light intensities and nesting records. So it would be these sectors that all belong to Saint-Paul, even if we saw that the maximum of luminance level were found in Saint-Pierre. Finally, we can resume our recommendation by this sentence, which is keep it low, keep it long, and keep it sheltered. So keep it low because by reducing the number of non-essential lighting, keep it long by using long wavelength lights, and keep it sheltered by reducing the proportion of lighting that are directly visible from the beach, and by creating natural screen and visual obstacles. So I'm not going to present the conclusion and future perspectives. So thank you very much for your attention.
minutes survey. Was the last the slide about which you want to tell something more?
I
It's
can
not
say?
a problem.
Okay,
Yeah,
perfect.
okay.
Okay.
It will be the 1 or 2 minutes less before the question.
Okay, perfect. So regarding our study results, we can say that it gives some interesting information for lighting management along the nesting beaches of Reunion Islands. And also we already organized some feedback campaigns during June to share the results. And so we so we organized this campaign by inviting different stakeholders, private and public, and we also organized this campaign with interdisciplinary collaboration with environmental association, public lighting managers and local communities in order to share the knowledge about this problematic and also improve as much as possible the local situation. For the future perspective, we can first say that it would be interesting to increase the community, the number of community outreach campaigns. Also to share the individual diagnostic and personalized lighting management measures that we already developed during the internship, which can help for as a decision support tool. And also it would be interesting to do a regular monitoring of this light pollution in order to see the evolution, spatio-temporal evolution. To go further, we can also integrate a monitoring of the study of the maritime propagation of the light pollution. It would be interesting because this can also impact at sea and not only when turtles are on the beach. Thank you very much.
I can still start. Thank you, Clémentine, for your presentation. I have a first question, which might be a bit of a provocation, but I like to provoke that.
Yeah.
Why did you introduce your presentation on turtles and not on other things? Because in the end, you worked on light pollution, which is a perfectly valid topic, but can affect pretty much anything. And as you say, turtle is no longer an ecological question in the Reunion Island, if I understand well. I mean, there are turtles, but they don't nest here.
Yeah. They don't nest anymore.
Yeah, anymore.
They
yeah.
still nest, but there is
it's one per year.
Yeah, it's very low. But the thing is, the association I was working on was really like focused on sea turtles. So it was important to have this first, this first focus on sea turtles.
Yeah.
Because we are doing different actions that are plantation of indigenous species, but also research about the habitat and everything. So we really wanted first to focus on this species, and also we were working with different organizations that were more focused on petrel, so seabirds, but also chiropter and other associations. So yeah, that's why also I wanted to film, but I know, it's...
But beyond that, there's a provocative and philosophical question, is that should we spend energy, time and money to favor nesting of a species that has stopped nesting here for like decades or even more?
It's a really interesting question, and I was sure that you will ask it.
And I'm sure I'm not making any friends behind me right now. I say
because when I arrived, it was also the question I was asking, like why we are focusing on the rehabilitation of nesting beaches when there is no nesting, so it just. And the thing is, the actions, especially the action of plantations, are not only for to create like. They are giving some interesting conditions for many things. So first, It will also so create this natural screen for light pollution, but it will also be interesting for erosion, beach erosion, and there is an important erosion of the beaches in Reunion Island. So there is, for example, some places where the houses are destroyed and everything. So to put this plantation will also improve, not improve, but decrease the rapidity of the erosion of the beaches. And it's important for also other species. And we see that through the action of this association, but also Colonia and other associations that work for sea turtles, it's not only nesting beaches, because there is a lot of juveniles and adult turtles that come to feed in around the surrounded waters, and so due to the different action that they did, there is more and more sea turtles that are coming. So it's also interesting to have this rehabilitation to see the potential impacts.
Okay, that leads me to my next question. I know those beaches, I've been there, and I know that the number of sea turtles have been increasing in the last decades in Reunion Island, especially due to the implementation of the reserve. So it makes sense that you want to favor nesting, because the populations are increasing, so you can expect them to nest here. Do you think artificial light pollution is the main threat, the main pressure that prevents nesting in Reunion Island? And what other sources of nesting failure or nesting avoidance could you list, considering the local context?
Yeah, so it's habitat degradation in general.
Yeah.
So it includes many different factors, but also the presence of urbanization, so of people on the beach. There is a lot of residents and touristic structure and stuff around. So of course it's also because there is a lot of noise pollution. There is also less and less vegetation, native vegetation in the beaches, and due to the coastal erosion, the width of the beach is shorter and shorter, and the frequentation of the beaches, I think.
Yeah. And well, I'm not a specialist of turtles, but yeah, I suspect that those threats, like having restaurants on the dunes, or having dogs wandering around, or having people lying on the beach, might represent some more serious threats. And I'm not saying it's not relevant to address light pollution, but I mean, especially for the outreach activity, I think it's important maybe for the people you're addressing to, that the hierarchy of stresses has to be very clear to them. It has to be very clear. As far as you know, this hierarchy, I'm not able at the moment to say what is the most important stressor. Okay.
Yeah. Also,
It's,
and the thing you said is very interesting, because I was talking about the sites, the sector that we had to prioritize for the lighting management, but for the plantation, for example, they prioritize the sites that have low level of light pollution and historical nesting records.
Okay.
So that's really the focus for this lighting management. Of course, first the site that need to be managed are in priority the one that have low level of light pollution.
Yeah. Okay, thank you very much.
Thank you.
Okay, thank you. So thank you for the nice presentation. I mean, one of my questions was related with the last one of my colleague, which is, I understand that light pollution can be relevant, but I don't see clearly. Maybe you can explain me how, because light pollution will be correlated with urbanization. will be correlated with coastal human activities and so on. So this is really what you are presenting here is a way to assess the effects of light pollution, or will be light pollution just a proxy to as an indication of anthropocized environments? Because I mean they are correlated, and so if you have more urbanization, you have more street lights, more activities, and if you have this, it's linked to coastal erosion and changes in the coastal management strategy and so on, and all this impacting in nesting and many other things that could be related to coastal ecosystems and so on. So, taking just light, do you think that is really you're really representing the impact of light, expected, the potential impact of light, or could we consider it just a proxy? Anthropogenic impact.
So for this study, it's the interesting point is that it's not just for it's not just to assess light pollution along the island, but it's also to propose some adapted management of lighting. So that's why it's important to consider lighting instead of anthropogenic factors. So first, we were thinking that it could be possible maybe to monitor through satellite images, for example, the light pollution that we would give also an idea of the distribution of light pollution and anthropogenic activities and everything. But the point is we want after to give some individual diagnostics of the lighting. So we did that. So for each restaurant, each shop, each structure and municipality, we gave some personalized diagnostic of the different lighting that we observed, of the different management possibilities that they had to reduce light pollution in these places, especially when they had nesting activity. And so in 2020, they found, they also did this study, and it was very interesting because in these sites, especially on the site number 2, they obtained an important decrease in the light pollution due to this study that had been done. And also people are getting more and more aware about this problematic. And so that's why we didn't study anthropogenic activity, but really we focused on
But I don't mean exactly that. I mean, if you tackle the problem of light pollution, you solve it and you design, and the problem persists, you discredit all the thing, because it's not the core of the problem, probably a sign of a bigger problem. It's not a general philosophical thing about that. But I think we should be aware of that, depending on how we propose the responses and the actions, it's not always beneficial, because you discredit the usual light. No, light is, together with other things, maybe indicating other things, or whatever. Because if you solve the problem of light, the problem is easy to solve. You don't solve the problems of urbanization or use of the coast zone and so on. And the problem persists, and they say, okay, we have done all this investment, all these changes, trying to get people aware of environmental conservation, and it doesn't work. So this is just a general, it's not related with you research, it's a general reflection about that. But the important thing for your research probably is the way of explaining it. So not to be too focused on light pollution, but light as an indication of something else, just in case.
Okay.
And one curiosity, just with this map, something I didn't understand in the methods. You were at the time referring to 23 kilometers, the proportion of 23 kilometers, and I look at the scale of 10 kilometers there, and the study area seems to be at least 80. Or what do you mean with the 23 kilometers?
So we didn't study all the coastline. We studied only
the 23 kilometers is the study transect.
No.
It's not the coastline.
No, it's not the coastline.
Yeah, thank you.
Okay, we have to stop here.
Okay.
Thank you.
Okay, so here we have Sarah Wurzel, so deciphering the lunar calendar of the brown algae. Sounds very attractive.
Okay, the floor is yours, Sarah.
Thank you for that intro. So I think I'll be in good company, but just to start, how many people know that seaweeds can reproduce with sperm and egg? Yeah, okay, very nice, very nice. This is something that I learned during this master's, the whole 2 years, but something that was really important for my thesis. And I'm specifically looking at this brown algae. This is Dictyota dichotoma in a low tide in its glory. We wanted to look at the lunar calendar and how it relates to reproduction. But we did more than that. So as a couple background knowledge, some important things that I needed also as background knowledge for this thesis. We looked at biological rhythms, which is related to biological clocks. And the thing with Dictyota dichotoma is it's not as simple as either an internal clock that is regulating how it does these rhythms that it has related to reproduction, but also some of its physiological outputs. But there's also external cues. And I'll explain a little bit why it's not easy to decipher which is more important or what the combo is affecting it. So for the purpose of our study, we were mostly interested on the external cues that would relate to monthly and daily cycles, but there are yearly cycles, such as season. So this is just a quick graphic that shows an example of some periodicities related to the lunar cycle. And in the case of Dictyota dichotoma, it's a circa semilunar cycle. So it's within 14 to 15 days that it has a cycle of release. So the life cycle. There are two phases. We focused on the right side of this graphic related to sexual reproduction. And so there are male and female algae. And it's a small photo, but these are male reproductive structures and female reproductive structures. These, you can only see a color variation, whereas with the females you can see distinct eggs. And this is related to the lunar cycle, whereas sporophyte release is less related to lunar cycle, if at all. So, my colleague did a great job of introducing light pollution. I'm going to refer to it as ALAN, artificial light at night. I had some examples of some recent and relevant literature coming out. Some of these are directly related to, or very close to, our study area, and some are related to trying to quantify it because it's going to be different at the surface versus something that's along the benthos, and different organisms. So one of these studies looked at coral gamete release, and then one is looking at oysters. Specifically, we know it relates to terrestrial and marine organisms. But we were interested not only that this is going to change how an organism perceives the lunar cycle, but it changes how they perceive their light-dark cycle, which are not exactly the same thing to these organisms and the processes they're involved in. And then we also were focusing on diffuse light pollution instead of a point source, which would be like a lamp, a street lamp that's on the boardwalk that's from a restaurant. We're looking at sky glow, which is something hopefully everyone probably relates to if you've been in an urban area and not been able to see very many stars. So it affects us all. So we had three main questions. The last one is more of a practical question. The first two were our main questions guiding our research. So we wanted to see if ALAN can desynchronize circa semilunar rhythms or dial rhythms in Dictyota dichotoma. And we wanted to see, is this desynchronization, if it's occurring, can it impact their physiological outputs? So not only reproduction, but other processes, which I'll get into later. And then these ALAN conditions, can we mimic them in the laboratory? If we see effects in the field, can we see those effects in laboratory conditions? Are we actually recreating this situation? So our study sites, this is a zoomed out picture, and then, great, I love that this board is big enough. So you can see, this is artificial light at night from lightpollutionmap.org. And our first study site is at the top, it's the White Star. This was Cap Gris-Nez. So here's a picture of the study site during the day from the tidal pool perspective. There's a couple buildings, but not very much. This is reflected in the light pollution. There's a little bit of light, but there's not a ton. However, if you go to Limahu, which is the White Diamond, there's a whole line of buildings. There's a lot of development on that coast, and there's a lot more light pollution that can be seen from that map. So we use this as our non-polluted or our control site, and this was our experimental or Allen site. We collected 10 male and 10 female gametophytes in each site, transported them to the lab. This was northern France. This part of the experiment was done in Ghent. So I took 3 pieces of phthalate from different parts of the thallus as well to get a better idea of each sample. And I put them in petri dishes with seawater. I kept these for 14 days because that's the length of this circa semi-lunar periodicity. So they were all in the control incubator. They were kept at 18 degrees Celsius, and they were, during the day, given light from LEDs, and at night they were given a full 12 hours. So they had 12 hours of light, 12 hours of dark as their day-night cycle, and this was kept constant. Each day, I moved the phthalate pieces, and with the petri dishes that either had the female gametes, the eggs, or the male gametes, with the females, I could use a stereoscope and just count them manually, and if they were too dense, then I did subsamples. And with the males, we used an imaging flow cytometer machine, which was amazing to count them, but it was in high demand. So I had to fix the samples and wait till I had the opportunity to process them. And this is a broad overview. It's very busy, so it's just to visualize both sites and both male and female gametophytes at the same time. This is preliminary data. However, the 2 darker lines do represent that non-polluted control site, and perhaps you can see that the peaks seem a little sharper and a little less diffuse with the light blue, the 2 light blue trends. There's more peaks happening, and they're not as sharp of a peak. So in previous data from the group that I worked with, the peaks should be sharp and distinct in areas that are non-polluted or controlled. However, this is also normalized, so with each phthalate, I did not have time to count the SORI numbers on each piece. So to adjust for the size of the peak based on how much was released, this was taken over the peak release of each sample during the 14-day period for each day, so that it was easier to compare the numbers of everything. And this, in this figure, this is the new moon in the natural cycle. It will be more clear in the next figure. So in the next figure, this is just male gametes from both sites, and I've been able to add the standard deviation bars because it was way too busy in the other figure. Once again, there's your new moon. These are the actual sperm, which is part of why it was awesome to use this machine, because you can get images. It takes a long time. And so these are the averages of 3 samples for each site for each day. Whereas, well, quickly, it's a little bit easier to see that there might be some variation in the experimental site, which is the light gray, and then the dark gray, which is the control site. For females, this is an average of all 10 samples. I was able to do that for the females. And once again, you have where the new moon is. This is a little busier than we expected. It's not quite as clear, but there still seems to be somewhat of, if not as sharp of a peak, maybe more of a synchronous peak. I will talk about that if I have some time. And I also followed lab samples. So the lab samples were prepared and entrained before I started my thesis. This is part of why we were interested in looking at the calendar. They can be entrained to an artificial lunar calendar in the lab. So it's not, they are taking external cues and using them to regulate when they release. So just to have them as a comparison next to our field samples, there was a setup with 2 different incubators. You know the control setup. So in the Allen setup, The goal was to make it more of a light dark, but to alter it. However, in practicality, it was a light light, but the light at night for those 12 hours was a reduced light, and it was with a different type of light, a tube light with gray film to make it more diffuse, and a softer light. But essentially they were in light conditions. I took 5 samples from each condition. I only followed females. I did not have fertile males to follow for the lab culture, which was one of the challenges in my thesis, but that's okay. And I counted them the same way that I counted the females for the field. And this was really unexpected results. So hopefully I have a little time to talk about it, but in previous results from the lab with entrained cultures, there is a clear trend in control samples. I was the first one to try to simulate Allen. And so for Allen, perhaps this is the trend. We had a lot of at least different signals with Allen, but for control, it should have been a clear peak. There should not have been low levels of release throughout the period that I was following them. And just one thing, this is the full moon that it received, so that is a mistake. It should not be dark, it should be light. Okay, so for physiological outputs, for the sake of time, I'm going to talk about gene expression. And we specifically looked at gene expressions that relate to blue light reception in Dictyota dichotoma. As far as I know, as far as what I've read in literature, that's the wavelength of light that they can perceive. So the process, in brief, we set up a time-lapse experiment. It was 24 hours. We sampled every 4 hours. And we started at when they got their first light of the day, which is an important cue for the algae, as it once again has that transition between light and dark. So at each time point, we had the 7 cultures that were in the control conditions, the 7 that were in Allen. We sampled 3 samples from each condition at each time point. We froze them in liquid nitrogen, and then eventually we processed them, we extracted RNA, and from that we got cDNA, and we put that in a qPCR machine, and we could actually quantify the expression of the genes throughout the time period we were looking at. So, very preliminary, but I'm excited about it. So, very briefly, the 0 time point, we had one previous study where a colleague had looked at BHLH and saw that it was rhythmic and also had a smile shape to the expression, which would be in the control conditions, which is the blue lines. So we knew that it should also match the 24 hours because those should be the same. That's the same moment where they get that first light cue of the day and they transition to a light situation versus dark situation. So we cut out the 0 hour for our time lapse because we weren't confident in those results, and in the future we would still need to repeat this to be able to see if this is a pattern in more than one time lapse scenario. However, this is the first time in Dictyota dichotoma that we've seen that there is rhythmicity in some of these genes and that there are some oscillations in some of these genes. Not all of them, but it's also interesting in that in some of them where there is rhythmicity, the Allen has either very muted rhythmicity or almost none. So this is a very exciting preliminary result, and hopefully says that yes, we were able to synthesize some of this condition in the lab. Okay, so briefly, the data may suggest that Allen can desynchronize circus and lunar cycles in Dictyota dichotoma. More research is needed to be able to confirm that or be able to say that definitively. I also want to do some analysis of that. And it also suggests that this could be affecting physiological outputs. So not only could this be affecting reproduction, which is very important for the species to continue and to continue in the places where it's found, but it could also be affecting other parts of the fitness of this organism if it's perceiving light differently and that's being affected. So yes, once again, first time that we're seeing the blue light receptor genes being shown in Dictyota dichotoma with rhythmicity. And briefly, related to Allen, it's interesting at first to be able to synthesize this in the lab, because if we have an idea of and a better control of their rhythms and we can manipulate them more closely, then we can do more lab experience related with Dictyota dichotoma and also related with and their physiological outputs. This could also be related to cultivation in the future of this algae and being able to have a stricter control of the cycle when we know what's controlling it more closely. And then just a little PSA with light pollution. It is one of the easiest forms of pollution to fix, and if it's affecting both terrestrial and marine organisms, and we see effects, then it's one of the easiest things as humans that we can cut out and help with some of the other stressors that are happening for marine organisms. So thank you.
Very much, Sarah. Actually, I'm sorry, in the last few months we weren't in touch because I'm very interested in this sort of matters. Okay, gentlemen.
It's me again. Hey.
I have the hidden one.
You're the hidden one, yeah. Nah, it's cool too. This is something that comes from my old friend Andres. We did our PhD together. Actually, it was 20 years ago almost.
That's awesome.
Yeah, I have. That's super interesting. But your results are super interesting. But I have some questions about the experimental not protocol, the design. Yeah, you're talking about rhythmicity, especially in the end with the gene expression results. The way I see it, you should at least follow the expression for twice the length of the cycle to highlight the cycle. What's your opinion about that? Because it's, I mean, my comments is also holds for the first part of your presentation.
Yes, so I agree. And actually the preliminary study was done over 48 hours. We chose, but it was done with less time points. Because our lab group, I was very lucky. I did this in Roscoff with Andres. I had a team. There was one point where there were only 3, maybe 4 of us, and there were time points at 1 a.m., 5 a.m. It's a tough thing. It's very cool science, but it is tough as scientists to study it this way. It's important. I think it would be good to do. I think I was glad we did the 4-hour time points instead of 6 or longer to get more details within the time frame. But yes, and especially because our 0 hour was off, it would have been really helpful to have another 0 or 24 hour, which is the same time point, to be able to say, okay, this is truly, we think we can support that this is the most accurate measurement at that time point.
Yeah, I think that's super important because I really enjoyed your presentation, but I was frustrated by that. I was like, yeah, okay, but like maybe she's just bullshitting me. and I know you're not. But
there's a point in being a
you see, I'm sure you experience the same frustration.
Oh yes, yes, yeah.
And I have another question. This time it's about the sampling design. Do your 2 sites, the one in Cap Grenet and the one in Vimbre, only differ by light pollution?
Are you referring to tides?
Yeah.
Yes, okay, great. So that is one of the main things. So this research, there is more research than just those 2 sites. For the sake of the time period of my master's, we focused on these 2 sites for this. Part of the reason why they are so close in longitude is because they fall within the same tidal range. So maybe they differ by minutes, but it's not along that coast. You get a variation. By the time you get to Roscoff, it would be hours. So that would be a huge difference in the cues that they're receiving. So this was planned so they're within the same tide.
Yeah, I know, but they differ by many other variables, like urbanization.
Yes. And it's interesting to think about that with light pollution and thinking about it as a marker of urbanization instead of just the
I think your remark was super insightful.
Yeah, I think that's very important. Also, that was part of why in the lab we were hoping to mimic those conditions, because then hopefully all you have is light as a difference.
Absolutely.
The temperatures are the same, the cycles are the same. Hopefully you are narrowing it down to light. but yeah, I think also the photos, it was really interesting to see even the amount of people. We did have a school group when we were sampling at Cap Green, so there were more people in the tidal pools at that moment. But otherwise, just in general, those regions, there's way more people in Vimeo, like way more people. And even the access to the beach has way more infrastructure so that people can get there. So yes, that has to have an effect. Even these Dictyota is also often in these tidal pools that when you have a huge variation, it's very easy to access them, these populations. So people can walk out and be amongst the Dictyota and be affecting it, yes.
Yeah. People, and I think even, if I remember well, Vimeo beach is like a huge sandy beach, but so we can expect, with like very exposed beach, so we can expect lots of sand scarring that can affect the seaweeds, while the Cap Green is more rocky, so probably less sand suspended. Well, but anyway, I agree with you that experimental would solve all of these aspects, and by making only.
Oh, I can't get back
light.
there. Yes, that was part of why that was. Oh well, that was one of our main things in the lab, was just trying to isolate that. But yes, if you want to see again.
Well, you don't see that, but in front of all those buildings, it's a huge sandy beach.
Yes, this part has
where all the region comes to the beach.
Sandy,
the weather is nice.
Yeah.
Thank you very much.
Thank you. Thank you for the presentation. It was very nice. It's a topic I like a lot. I was for years working on circatidal tissue renewal in mollusk, semilunar, and these kind of things.
Okay.
When I was younger.
Okay.
Younger, not young. And this, my mind criticism or people was related with the aspect that to get a cycle you need to repeat at least 1.5 times or 2 times.
Yeah.
And I know, I appreciate what you say. I know that you need a lot of effort, personal effort, and to follow that.
Yeah.
You have Urzi around there, which one of the summers. We were doing some experiments collecting samples every hour for 3 days, non-stopping. And the only thing that means is that you need to forget about everything you want to measure, focus on the specific target that is simplistic, and with this you can track transversally in order to compare with the more details. So I would suggest that it could be an option to think on simple endpoint that you can make there for 2 repeats, maybe not 1.5, maybe 3 or 4 cycles.
Yeah.
I thought that the gambit release was a good opportunity for that. And maybe you can make it longer and see. And only then you can see whether you have this cyclicity.
Yeah. With the lab samples, one of the major themes of this thesis is that culturing algae is hard. And they don't always behave, in fertility especially. That window is hard to make sure you get, where you get your cultures in the same window of fertility. But yes, I agree. The goal originally was to follow it, at least in the lab, over multiple cycles. But realistically, I didn't have the fertility or the consistency.
You can simplify everything else. You know, focus on the repetition of something.
Yes.
Could be.
Yeah.
And the other methodology question is you were using, you have mentioned, if I am right, bright light in experiments in the lab with a reduced spectrum. Have you tried anything of trying just using the very narrow blue spectrum? Because when you are in nature and you're in depth, after 1 meter, you're going to have blue, and this is the one affecting the cell turnover in corals, hormone regulation in many invertebrates, and probably also gamete releasing in algae. So
yes.
Is very narrow blue, or it is wider, the experimental one you had used?
The experimental one we had was white light, yes. So it was wider. I think that would be a very interesting study, as well as the different levels. We kept it to the same level. I had experimental conditions in Ghent and Roscoff, so I measured them and adjusted them to be comparable, but we had 1 level. And one of the other studies had a series of levels, and the lowest one was close to what Clementine already said, mimicking that full moon level, which we were well above. So I think in the future it would be really interesting to see, okay, if we do have effects at this level, what level do we start seeing effects? What is the amount of lux that we start seeing the effects? But I think also, yeah, the range would be interesting. Yeah.
Okay, so that's all. Thank you.
Yeah, yeah.
Congratulations.
Okay, well then I have to say, I have to say,
or maybe you have other questions?
I'm happy.
No, no, okay, okay. Okay, well, I have to say something in sense that all this is super interesting to me. Okay, to put I mean this project in the context, it's important to say that okay, of course, Alan, those artificial light at night, nowadays is a topic that is receiving a lot of attention, and by now there is, I would say, a decent amount of literature available, but almost everything is actually on animals, or the land plants, or about the photosynthetic organisms in the sea. There is essentially zero, basically zero. I am aware of only the two very recent papers, one on Posidonia oceanica, published by colleagues of the University of Pisa in Italy, and another one in which the seaweeds were a secondary part of a study carried out in the Sydney Harbour. Okay, so let's say whatever the new data come out, delivering some information on this are super valuable. Actually, I should say that also there is a paper submitted at the moment. And then, okay, when you were in Ancona, Sarah, did you ever go to the Passetto at night?
I think I did once, yeah.
Okay, have you ever seen it?
None of your business.
Okay, you should have seen already the Fantine, the Zoe, okay, you should have seen that there are stretches of the shore which are more or less in the dark conditions, and the other stretches that receive a lot of light. Well, okay, a couple of months ago, based on MBRC thesis of the girl who graduated a couple of years ago, the British girl, okay, and we submitted the manuscript. This is actually a very simple thing. We compared the communities growing in the exposed spots with the communities that grown in the sheltered spots, and also okay, in a place that in a big rock pool where there is a population of the Gongolaria barbata, which is partially exposed to light and partially shaded, we made a comparison about that. So we don't have the type of data that you have, but I mean, this is okay. Okay, I will let you know how we will receive the response from the journal.
Okay.
Look, this is very interesting. Getting back to the point that Gauthier remarked that that's okay. There is an aspect that should be taken with some care, because in your case you have one site exposed and one site sheltered, so as the control. So in principle, what Gauthier says is right. I mean, the differences that you find them might be due, in theory, to even to other factors or something which is not directly related to Alan. But still, once again, let me stress that whatever the new data we get on this topic is extremely useful and valuable. So okay, I'm just silent. Okay, I'm very happy to see now what they
We understood that.
Any other
You
questions?
have examples with coral synthetics or Santella.
Okay, nice.
Barcelona in the weekend, and it was raining cats and dogs.
Davey, are you ready?
Yes, I'm ready.
1 minute left.
Okay.
Yeah, Maren. Yeah, they're sending a message. Maren, it's way. Maren. Okay. Okay, welcome everyone to this last session of today. Last but not least. Don't get scared because we are five now in the tribunal. So a lot of people to make questions. But don't worry because I will try to do that and to avoid using more time that is assigned. Okay, so remember all the people that will present that you have 15 minutes. Okay, when 2 minutes left, I will say. That means that. Okay, in the tribunal we have Aldin from University Cote d'Azur, Gonzalo from the University of Oviedo, Laurence from University of Sorbonne, and Maren from the University of the Basque Country, and I am Gonzalo from the University of Oviedo. You have 15 minutes, it's your moment, on the floor, it's yours.
Thank you.
Go.
Hi everyone, good afternoon. I'm Gibbs Samuel David, and I'll be presenting my master's thesis carried out at Atlantic Technological University, Galway, Ireland. My thesis is titled Histopathological Assessment of Blue Mussels, Mytilus edulis in a Contaminated Salt Marsh and a Contrasting Good Water Quality Bay. So how close are you to the coast? Chances are within 100 kilometers. This proximity to the ocean is the reality for about 40% of the world's population, thereby placing massive pressure on our vital ecosystems. And salt marshes are important ecological, and there are important vital ecosystems that provide ecological and functional benefits, such as carbon sequestration, habitat for seabirds and the likes, and also food, and even they support also coastal erosion. But despite these huge many benefits, they are being threatened by urbanization, pollution, and climate change. However, there has been legislative procedures set in place to manage them, such as protected areas, manage realignment, and salt marsh restoration. Anthropogenic pollution, particularly chemical and biological contaminants, threaten marine ecosystems and human health. This is because pollutants bioaccumulate in marine organisms, and thereby they also magnify through the food web, and thereby posing threats to human health. And salt marshes are exposed to plastic litter, especially salt marshes in urban areas. They expose them to plastic litter, microplastics, trace and heavy metals, and other contaminants. Blue mussels, Mytilus edulis, are one of the vital marine organisms found in salt marsh. They are very important in Irish economy, as they provided about 17 million euros in export alone in 2021 to the Irish economy. And they play a crucial ecological role in salt marsh ecosystems, and they are definitely impacted by anthropogenic pollution directly and indirectly. So understanding the health of blue mussels is crucial in assessing environmental risk, ecosystem health, and also potential contamination transfer in the marine food web. One way to look at this is to assess histopathological alterations in vital tissues in mussels and in a salt marsh environment by comparing them to a cleaner site. So we set up to ask a research question, which was, do blue mussels from an urban salt marsh with known microplastic pollution and anthropogenic stressors exhibit histopathological alterations in their gills, guts, or gonads compared to mussels from a clean site? And then to address this question, we set out two objectives, which were to assess, one, the condition index of mussels from both sides, and secondly, the histopathological alterations in the gills, the guts, and the gonads from both sides. Our hypotheses were thus that one, condition indices will be better in muscles from the clay site, and also inflammation response will be less severe in the gills, gut, and gonad from those from muscles from the clay sites. So these were my study area. To the left is Galway Bay, which is also the clay site and also known as Site 1. And it's located on the west coast of Ireland. To my right is our salt marsh of interest, which is Butterstone Marsh. It's a natural reserve and it's located on the east coast of Ireland, in Dublin in particular. It's a polluted site and also our site too. Based on human consumption of mussels, the European Union Shellfish Water Directive classifies mussel harvesting areas into Class A, Class B, and Class C. For Class A, they don't need to undergo the mandatory depuration. Depuration is when you place mussels in holding containers for at least 48 hours for it to deguss some of those particles or contaminants it has ingested. For Class B, you have to leave for at least 48 hours, and then for Class C, you have to leave you have to take it to heat treatments for it to meet Class B or Class A. So our Site 1 was Class A mussels, while our Site 2 was Class C mussels. And then so we collect for those in Galway Bay, we bought them from a local market in Galway Bay, and they were road cultured mussels. While those from Butterstone Marsh, we collected them from the natural sediments somewhere around here. So in total, we collected 72 individuals, 36 from each site, and from this we collected data on the biometrics and also extracted the gill tissue and the gill gut and gonad. However, for the purpose of this study and then also due to time constraints, we randomly selected 20 individuals, 10 from each side. Recall we already collected 3 tissues from each individual. This gave us 60 tissues in total. And from each tissue, we collected micro sections or slices on 10 glass slides, giving us 600 glass slides. So at the end of the day, I was working with more than 600 glass slides, which was a lot of work to do. So this was how I assessed my condition indices following the methods of Hammer et al. 2008, and then those were how I measured my biometrics. For the statistics, I used Shapovalov test and Levin's test to check the normality and equality of variances. Having passed these 2 tests, and because they were just 2 sites, I had to use 2 sample tests because 2 sample t-tests because they were areas that were independent of each other, and then my alpha level was set at 0.05. So welcome to this wonderful maze, which I won't go into time to explain, but this is your typical histological technique. I adapted this method from Costa 2018, just beginning from your tissue extraction and then fixation down till your macrotomy with your rotary microtome, and then your staining procedures down till when you view them with your microscope. However, this was the area where I adapted my methodology. It was mostly the timing. So I adapted this timing from Costa et al. 2018 just for the tissue staining process. Yeah, results and discussion. Our shell length range between these, and then they were within the range recommended by OSPA 2018 for either for mussel consumption or for mussel studies of this type of study. And then it's important to state that while we were doing our extraction, we did what we call presence and absence of parasites by naked eye observation. And then it was shown that about 11% of our samples were positive for parasites. And then in terms of sex ratio, we had 13 females and 7 males. And particularly for Site 1, the ratio was 1:2 for male to female and 1:1 for male to female. Yeah, so was there a difference in the biometrics between the mussels from Site 1 and Site 2? Yes, we did have a difference. As we can observe here, the total weight and shell weights were higher in Site 2, while the other biometrics were higher in Site 1. I'd love to state that the green bars denote site 1, while the orange bars denote site 2, and this carries along the rest of my presentation. Yeah, so did we have a difference in the condition indices for both sides? Yes, we did. As we can see here, all the condition index assessed were better for site 1. Coming down to the histopathology, was there a variation in the inflammation observed for the gills for both most cells from the two sides? Yes, we did have differences in variation in terms of inflammation. As we can see here, this is how a normal gill should look like, and we got this from site 1. And this is how an inflamed gills look like. We got this from site 2. As you can see, swelling in this region, the frontal zone. In principle, site 1 was less impacted than site 2, 25% compared to 90%. We did observe other forms of inflammation also, all coming from site 2, as we can see, epithelial disruption down to presence of hemocytes. We also looked at the gut, so we also observed differences in terms of inflammation for both sides for the gut. And then, as we can see here, this is how an inflamed gut looks like. We got this from site 1, and this is how an inflamed gut also looked like from site 2. So in principle, site 1 was also less severe than site 2, although a bit higher, 40% compared to 90%. We did also observe other forms of alterations. As we can see from site 1, we got digestive tubule atrophy. From site 2, we had something like lipofuscin and hemocytes. We also couldn't identify some of these items which could be undigested and food particle being found there. But we think we suggest that looks like Daphne, especially for this. Yeah, so was there a difference too in the inflammation in the gonads for both sides? Yes, we did have a difference. And as we can see here, this is how a male normal gonad should look like, and this is how a normal female gonad looks like. And then we also had some other alterations. No, before I go there, interestingly, site 2 was less severe than site 1 for the gonads. 50% compared to 66. I'll come back to why this happened during my explanation here. So we had some other forms of inflammation. For instance, from site 1 we got oocyte atresia, and then from site 2 we got stuff like gonad degeneration and follicle degeneration. We found out that for site 1, they had better condition indices and minimal inflammation in the gills and in the gut, while for site 2, we observed that they had poor condition indices and pronounced inflammation in the gills and in the gut. However, for the gonad, they were more or less the same, except that it was more severe in the females. Studies suggest that females are more prone or are more susceptible to contaminants because they expend more energy during the production. So that's why I feel like we had more. That's why I feel we had more inflammation in site 1, because it was 2-to-1 ratio female to males for site 1, while for site 2 it was 1:1. So that's how we hypothesized that it was because of that. These inflammations, yeah. So these inflammations, we think these inflammations for site 1, we attributed these inflammations to natural processes, parasites, and also macroplastics. Why? So our samples were collected at the end of January and at the beginning of February. Studies have shown that most cells spawn, or they have their spawning period between April and the beginning of January. So getting our samples at the end of January and the beginning of February means that there will be lack of stimuli for spawning. And in that case, when there's a lack of stimuli for spawning, naturally the ripened gonads would degenerate, and follicle and male gonad degeneration were the main forms of inflammation we observed in our study. And for the parasite, like I stated already, we had 11% of parasites. That is just by visual, by eye observation. So we propose that if the other 89% is being put under microscopic investigation, there is a high chance that they would have parasites. And then also for the microplastics, yes, my work is part of a larger work, and then we've seen the lab has already studied microplastics in this GAWB for the mussels, the sediment, and also in the water. And for Site 2, we link this to microplastics, PCBs, PHs, contaminants of emerging concerns like paracetamol. Microplastics also too has been assessed in this salt marsh from this lab, and also we have a train station just by the salt marsh which might be releasing oil into the environment and thereby introducing stuff like PCBs, PHs, and all these are linked to poor water quality. I would love to say that we use the European Union Shellfish Water Directive to differentiate between both sites, and we saw that Site 1 had better water quality, particularly for the dissolved oxygen and for total suspended solids. What does this mean for the mussel health? It means poor growth, reduced feeding activity, impaired respiration, poor reproduction, and high chance to diseases. Perspective for future research. We encourage future research to look at what toxins are causing this disease specifically, because some of the older buildings in Butterstown is not connected to the central sewage treatment work, so they release untreated sewage into the North Stream, which feeds the Butterstown marsh. So we think there might be toxins being introduced, and we did not assess this. So future studies should look at this, and also looking at carrying out microscopic investigation for parasites, and also adding biochemical biomarkers to study this and to corroborate our results. In conclusion, our results are consistent with recent studies which have shown different xenobiotics to cause inflammation in mussels. Their studies were done in the lab. Mine was done in a salt marsh, but I still observed the same form of inflammation. And our hypotheses are accepted because they are in line with what we had proposed. And the major takeaway from this is that the conservation strategies already put in place for this salt marsh should continue to be enforced. These are my references, and these were the common words found in my research, and thank you for listening.
Thank you so much, David,
Thank
for
you.
your presentation. Is there any short questions in the audience? Very short, because we are 5 now. No? Okay, so Aldin, do you want to start?
Yeah, okay. Thanks a lot for this really nice and dynamic presentation. So you were talking a lot about this pathology or atrophy, or so. How do you define in your muscle this atrophy, or I mean how? What makes you say that it's this specific phenotype that you are looking, it's atrophy, and this specific phenotype, it's there is an alteration, and what does make you think that it's pathology and not physiologic, like after spawning? I'm sure that the gonad retractor get another shape, so but this is physiological and not pathological. So what makes you say that?
Yes, this was one of the constraints for our work, because we did not assess specifically what toxins are there due to time constraints. But from what we've seen in the environment already assessed, like we got water quality data from Dublin City Council, which looked at the nitrates and other pollutants. We also got data looking at the Shellfish Water Directive. So looking at this data and also looking at what has been done too in the salt marsh, like we've seen plastic litter, we've seen untreated sewage into the system. So we attribute that these are connected to these toxins being introduced. And then at the other end, we also feel like sometimes it could be natural processes. Like we couldn't see if parasite played a huge role because we did not do macroscopic investigation for parasites. But we would think that parasites could also play a role in this, and not just other toxins.
Okay, then what would be the control for your experiment to really convince that?
Yeah. So the control was the clean site. For the clean site, yeah. So for the clean site, we had better results, like less inflammation in the gills and in the guts, just only for the gonad, which I explained our reasons why we had those inflammation.
Thank you.
Thank you.
So then just one more question. So you were talking about inflammation. So what are you calling inflammation in muscle? What makes you say that?
Okay.
That there is inflammation.
I mean, inflammation could occur when there is an introduction of a foreign body in a system. For instance, there could be infiltration of hemocytes, which are response to diseases. So once you see those things in quantity that is higher than normal, that's an inflammation. And also for stuff like lipofuscin, which are breakdown products of lipids, when you see them, there are also signs of inflammation. Yeah.
Okay, thank you.
Thank you.
Thank you, Jane. Good.
Yeah, so thanks a lot for such a nice presentation.
Thank you.
So I have one main question, because you are using a control sampling site, but it's really a control. It's like, have you thought about characterization of different kind of pollutants, such as microplastic in that particular region? Did you take some water samples there, or it was already known? Or?
Okay. So, like I mentioned earlier, we used the European Union Shellfish Water Directive, and for this directive, that particular site, Galway Bay, is classified as Class A.
I heard that, and that was clear for me. But what I'm saying is, like, even for example, the content of microplastic when you are inserting could change from one season to the other sometimes, and the composition of microplastic or other kind of pollutant. Apart from that classification of control, did you have a quantification of the impact by different? No.
And not for this study, but as part of the larger work, we have also assessed the level of microplastics in Galway Bay, which is less more than the number we have at Butterstown Marshall.
Okay, I will include that number. Just apart from the characterization of A, B, and C, I will show that number to make it clear that you can clearly use as a control. And just some suggestions. So,
okay, thank you.
And it's possible. Do you know if how different are the population? Can you do some sort of transplant experiment where you can see if that thing that you're observing when you go from the control population with some cages or whatever you put in the not so clean, let's say, region, if you are coming back to this kind of pathological thing? Have you thought about it? Is it possible? Is it feasible? I would like to know your opinion.
That's one way. That's one thing we are looking at if we are to push this work further. Yes, but due to time, this was what our scope would carry due to time constraints. Yeah, but definitely we can do that and even much more than that. Yeah.
Thanks a lot.
Thank you. Thank you.
Congrats.
Thank you, Laurence.
Yes, thanks for your presentation.
Thank you.
A lot of histological work, and so I have some question maybe that is similar to what my two colleagues have asked already. But regarding this inflammation process that you described, I am wondering if you were able to see a trend like in the 11% of organisms that were infected by the parasites. Was there a trend that those organisms had more more inflammation, or
no, there was no trend,
or more hemocyte? Neither.
No
trend at all.
Yeah, there was no trend. Yes, I looked at the data, just scanning through, looking at the parasite, the ones infected by parasite, and then I looked at the kind of inflammation. They were similar to even others, so there was no trend. Yeah, correct. But we that was just visual eye observation. If we could go deeper, then there's possibly that there would be trend. Yes.
And those parasites, they were as frequent in the clean side as in the other side?
The parasites?
Yes.
Oh, the parasites, interestingly, the visual observation was the 11% we actually had were all from the clean site.
Okay.
Yeah, interestingly, yes.
Okay.
Yeah.
Thank you, Laurence. Martin.
So, thank you for the presentation.
Thank you.
Congratulations for the work that you have presented. So, I have a question about the prevalences, because I think that at some point you mentioned, but I haven't. Okay, maybe it's because I was not able to see that part of the.
Yes, I did mention that.
Okay, so
Sorry.
this prevalence, how you calculate the prevalence?
Okay, so I do this. We pick a muscle, and the reason why I worked with 600 slides was so that we could exclude the possibility of not seeing something exactly. So that's why I had to collect 10 glass slices per tissue, so that even if from one tissue from one slide we may not see what is there, other slides might show us something. So what I did was picking 10 slides from one tissue. Once you observe an inflammation in one of the slides, that means that we have positive for inflammation. So that gives it the percentage. So looking at all the 10, okay, let's say from site 1 we had 300 slides. So the 40% means that about 40% of these glass slides showed inflammation. Like 10 slides per organ. Once I have one inflammation, the whole slide shows, for that particular organ, it means that I have an inflammation. I go to the next organ, I look at the 10 slides. If I don't have any inflammation, it means that for that organ, no inflammation. So that's how I use it.
Okay, so that means that you don't calculate even the severity in each of the samples.
No. It's something I intended to do if I had more time. I wanted to use softwares like QPath, but there was no time for that. I also wanted to use what we call histopathological index to calculate it, but that's not very possible for 600 slides. Yeah, from studies I saw, they were using like possibly 10 glass slides, 20 glass sets to calculate that. It's very efficient. You can look at the severity, classifying each severity of inflammation and giving it a number within 0 and 6 or within 0 and 3, and then do your index. But for 600 glass slides, for a master's thesis, it was impossible for me to embark on. But it's definitely something I would love to do if given the time and the time it requires.
Because for some of the pathologies that you show, I mean that it's also important to have the severity, because there are, as you mentioned, there are several natural conditions that can at some point modulate some of the responses. So depending not only the prevalence of the individual showing, but also the severity gives you a more in-depth, let's say, a more robust.
Yes, exactly.
And did you check differences between sex in the pathologies and in the prevalences, or you haven't considered that?
Oh, for the sex, we had more prevalence.
Not only the gonad, digestive gland, for example.
Okay, like males as a whole and female as a whole, both for the gut, both for the gills and for the gut.
All the pathologies that you have shown in digestive gland.
Okay.
So are more dependent in one sex than in the other, or there is not?
No, where I considered sex was only for the gonad, not for the whole study. Yeah, where I considered sex was just for the gonad and not for the study. That was because when we were extracting our data, we didn't delineate which ones were male and female at that point. It was during visual observation with our microscope was when we can tell, okay, these are males, these are females, and then we were able to segregate them. But just based on the gonads, not for the whole work. Yeah.
Okay, because I know that there are some pathologies that are more, that you mentioned, so the gender can.
Yes. But several studies have shown that females are more sensitive to pollutants than males because of the kind of energy they expend during reproduction.
And one last question, if possible. So the sample, when you collected the sample, I mean that you dissected on site, you brought the muscles to the lab and then dissect them in the lab.
So we brought the muscles to the lab and dissected it there the same day. Like for instance, for the one we got from the local market, we went as early as 7 a.m. If I remember, we went there, collected them, and then once we arrived, it wasn't that far from the lab, about 10 minutes drive. Once we arrived, we started dissecting them. For the ones from Butterstone Marsh, we after collecting, we put them in a moist container with some kind of soil, and then brought them to the lab. That same day, we dissected them. Yes, so they were all alive when dissecting them.
No, it's not a matter of alive. I mean that more in the case of the gonad. So depending the time that it took until the dissection,
Okay.
sometimes, so they start like an spontaneous spawning and release of the gametes.
Okay.
And in some of your pictures, what I saw is that this could be one of the side effects of the process. For example, the upper one is spawning
This one?
on
This one here?
Yeah.
Okay, follicle and degradation, yeah, okay.
I wouldn't say that it's follicular degradation. It's more that it's releasing because the express situation the gonad, and maybe this is because the transport or the manipulation.
Okay.
Could be, from my impression. Here you have hemocytic infiltrations.
Yes.
It's very clear, okay. Yeah. But in the upper two pictures, you have more spawning because the manipulation or maybe the time that it takes until dissection or whatever.
Okay, thank you.
Okay. Thank you very much.
Thank you.
Okay, thank you so much, David. You are done. Eureka. Are you ready?
Yes.
Go ahead.
Good afternoon. My name is Nova Yurika. I'd like to present my thesis project entitled Identification of Transcript Encoding Enzymes Involved in the Synthesis of Anti-Cancer Compounds in the Diatom Chilindrocella clostridium. I conducted the thesis in Italy, Stagione Zoologica Antodor, under supervisor of Dr. Chiara Aurifano. So the microbiology or the photosynthetic microorganisms serves as the basis of the food web and because of the primary producers, and they are available in high abundance and high diversity in the oceans, and they can be easily cultivated with low cost and simple methods. Under biotic and abiotic stress, such as nutrient deprivation or addition, this microbiology can change their biosynthetic pathways and produce the secondary or primary metabolites, which leads to the attraction of several biotechnological applications, such as a new source of biofuels, nutrition, or pharmaceuticals. And with increasing numbers of cancer cases worldwide, it's also important to explore the microbiology potentials to be used as the for anti-cancer compounds, specifically as the photosynthesizers in the photodynamic therapy treatment. Okay, so what is the photodynamic therapy treatment? So it's basically emerging and less dangerous cancer treatment based on the administration of the photosynthesizers and its activation by certain wavelengths correspond to the wavelength of the photosynthesizers. And after it's activated by light, it will react with the oxygens and release the reactive oxygen species, which eventually kills the tumor cells in various of ways. And in my thesis, I focused on two molecules of photosynthesizers. First was aminophenolic acid as the precursors of the biosynthesis of the chlorophyll. And the second one is fluorophorate A as the degradation product of the chlorophyll. And microbiology has really big potentials in the production of these molecules because they are photosynthetic organisms and really abundant in the oceans. And in the microbiology, they are involved in the porphyrin pathway. So my thesis was only focused on the identification of transcripts, meaning in the transcriptome, encoding enzymes involved in the synthesis of these anti-cancer compounds. And so the Chilindrocella clostridiums were grown into nitrogen condition. The first is a nitrate medium as a control, and stress conditions in the urea medium. So the idea is after the cultivation of this microbiology, I also aim to assess the genes of expression analysis to see in which conditions our genes are upregulated, whether in the nitrate medium or in the urea medium. And my hypothesis was the genes of interest are upregulated in the urea medium. Moving to the methodology first, before the thesis, the transcriptome of the Chilindrococca clostridium was available, its sequence annotated and assembled. And during this thesis, I started to look for the gene of interest by looking at the porphyrin pathway in the Kyoto Encyclopedia of Genes of Interest, or KEGG. Because of unavailability of the pathway of my microalgae, I used another microalgae, Phytopterum trichordatum, as a model species in looking for the pathway. So once I get the list of gene of interest involved in the pathway, I did the transcriptome analysis manually by looking at the transcriptome data of the Chilindrococca clostridium. And if the genes of interest was available in the transcriptome of Chilindrococca clostridium, then the sequence are validated using BLAST. For the candidates of reference genes, I took some study for the previous studies. In the next step, I designed the primer. I cultured the Chilindrococca clostridium in triplicates in two groups, nitrate condition and urea conditions. And once the microalgae reached the exponential and stationary phases, I isolate the RNA and did the quantity and quality check of the RNA. And then the RNA then transcribed into the cDNA. Then the cDNA are used for the real-time qPCR analysis. Then after getting the CT value of qPCR, I did the reference gene assessment by RefFinder using 3 different softwares. And finally, the gene of expression analysis by relative expression software tool or REST tools. And I did the statistical analysis by the GraphFact. The results are divided into 4 sections. First, we will see the result of the genes of interest. Okay, in these figures, you can see the pathway map of the genes of the molecules. First is aminophenolic acid, shown in the upper corner of the map, and the second is phenobarbital A in the down corner of the map. I apologize for the quality of the pictures. I captured it from the KEGG website. But overall, in the green boxes, you can see the genes of interest. There are 12 genes of interest identified in the transcriptome of the Chilindrococca clostridium. And okay, this is the gene name in the model species Phytopterum trichordatum and the enzyme code. And the abbreviation of the genes, and the genes, the data was from KEGG, and the genes name in the Chilindrococca clostridium from the transcriptome of the Chilindrococca clostridium. Okay, moving on to the reference, the candidates of reference genes. I have 4 candidates from the previous studies. First is tubulin beta, GADPH, and actin and EFL. For the RNA quality and quantity check, the ratio of sorbents of 2060 and 2080 nanometers shows the value bits around 2 that indicated that the samples of RNA were pure, while the ratio of sorbents of 260 and 230 ratios, some are below 2. We conclude that this is because the presence of the co-extracted contaminants, because we use the triazole reagents that have the quite similar absorbance with the RNA. For the reference gene assessment, we have 3 softwares. We use 3 software. First is BestKeeper, that assess the best reference gene based on the lowest standard deviation. Here you can see that the most stable genes or have the lowest standard deviations was tubulin beta. The lowest standard deviation is the better, the more stable is the genes. And for the norm finder, which assess the stable genes according to the ANOVA analysis, show that the tubulin beta was also the best reference genes here. The lowest the stability value is the better for the reference genes. And according to the genome that use the pairwise variations, to calculate the maximum number of reference genes, it can be indicated that the combination of tubulin beta and GADPH was the best reference gene. So based on of these three results of different software, we took tubulin beta as our reference genes for the real-time qPCR analysis results. Okay, moving on the last result here, the REST tools and statistical analysis shows that growing the Cylindroteca clostridiums in the urea stationary conditions show the higher gene expressions, result in the higher gene expressions compared to the control stationary conditions or natural conditions shown in the x-axis. So the x-axis is a control and the up one is the urea stationary conditions. The result will show in the logs twofold expression ratio. And we also assess which phase is the microalgae can express the higher expressions of gene of interest, whether is it exponential or stationary, because from the previous study, the molecules of interest were produced more in the stationary. So we want to also know whether is it in the case of our microalgae or not. So the result here show that growing the microalgae under the stationary phase will result in the higher expression of genes of interest compared to the exponential phase in the x-axis. So the result will also shown in the log x twofold expression ratio. So this is my thesis was the first study that assessed the transcriptome of Cylindroteca clostridium to see its potential to be used as to see the transcript of the microalgae to be used as to produce the photosynthesizers molecule of anti-cancers. So we show that tubulin beta is the best reference genes for the urea stress cultivation. Actually, the result of reference genes varies depending on the species or the organisms that studied, the environmental conditions that were lived or grown, and also the developmental stage in which the microalgae were at that time. And also, we conclude that the trend of upregulation of all of the genes of interest can indicate that the two molecules, fenobarbital A and aminophenolic acid, might be produced in the higher condition if we grow them in the urea medium and cultivated them in the stationary phases. So we also conclude that our hypothesis is accepted. But we want to note that this thesis only provide the information of identification of transcript. I mean, we only provide the transcript in the Cylindroteca clostridium. So why we do that? Because we want to serve it as the basis of future genetic engineering. For example, to identify the target genes to be modified in the future for the higher maximum of yields of these molecules. And so, for the future perspective, we really hope that the next researcher or I can do the chemical analysis to confirm if there will be higher productions produced in these conditions, and to extract the fenobarbital A and aminophenolic acid, and also the bioactivity of in vitro analysis in the cancer cells to confirm, and also the genetic engineering to modify the target genes, modify the target genes for the production of higher compounds. Okay, that's it. These are my references. I'd like to express my sincere gratitude for my supervisor, Dr. Chiara Lauritano, and my lab mates, Eleonora Motori, and also my family and friends. And thank you very much for your attention.
Okay, thank you so much. This time we will start by this stream, so
Thank you, yes. So thank you and congratulations for presentation and the nice work that you did. And especially because I think that culturing diatoms is not so easy, especially growing them. I don't know how easy it was for you and just maintaining this kind of culture for your experiment.
It's depend on the species. For my species, it's called Cylindritica closterium. I have easy methodology and easy time to produce, to grow them. So yeah, I don't have any difficulty.
Okay, so how long, for example, until you get, I don't know, if you go from the through the exponential phase until you...
1 month.
In what?
Yeah, in 1 month.
Okay, but it's your success. So your interest in this species, okay, the thing is why? I mean that why diatoms to obtain this kind of, in this case, genes, but with the aim of, as you mentioned, to produce this kind of chemicals in a later stage. Why not other, let's say, more simple to use algae? Or
because diatom is the most diverse group of the microalgae. So, and also they are pretty easy to culture. And I want to show that, especially for Cylindritica closterium, and I want to show that with my result now, it can be like the basis line for the future study to use another diatom, yeah, to get the similar, and to get the compound. And especially I think that the diatom is really have big opportunity and to grow this, to produce these metabolites because they are really fast and abundant in the oceans. Yeah.
And from the results that you show about the when you compare the transcription levels of your genes, you know, it's just comparing with what is in literature, and so that's really is a relevant high expression. I mean that makes this species suitable.
Sorry? How?
So, for example, the transcription levels of the genes of interest that you show that, okay, it's statistically significant and blah, blah, blah. But that is, I mean, that these fold changes are so relevant in order to choose this species for future studies in production. There are some others, maybe that respond more or less. It is true that you are comparing culture media, okay, but because I saw that, I can understand from that you have like
in comparison with other potential sources.
Yes, I see that in that sense, you have 3 times more, no? I guess.
Yeah. Yeah. Usually the diatom will grow in the nitrate condition. So I just want to explore what if we use another nitrogen sources, which is the urea. And I got surprisingly better result if we use the urea in this case, because all the genes are upregulated, means that there is a higher chance that the metabolite will be produced more because the genes are expressed more in this case. So yeah, that's why I did that.
Thank you.
Thank you.
Thank you, Maren. Laurence?
Yes. Thank you for your presentation. I have also some question about why you picked up such a culture condition. Like, for instance, why did you choose urea and not ammonia, for instance? And then the second question is because it's the genes are in the photosynthetic system. Why didn't you play with light instead of with nutrients? What was the reason for this setup?
Yeah. So for the first question, why I choose the urea, because I did research from the previous study. They also studied that the urea also show by using the urea medium. It also show the increase of the expression levels in the Diatominotocolus polymorphus. They not directly studied the pathway of the molecules of interest, but they studied the porphyrin pathway. So yeah, I just want to confirm, is it in the case of result, the same result in the my microalgae as well. So that's the first thing. That's why I use the urea. And for the second question is
why didn't you use different light condition?
Yeah, because first I would like to really do that as well, but given the limitation of the time of the master thesis, it was like around 5 months, so it was not possible. But in the future, there is like a lot of possibilities to do. For example, the light and the chemical analysis and the extraction of the molecule of interest. Yeah, I would like to do that. But it's possible to play with light as well.
Okay. Yeah, so thanks a lot for the presentation. So about this, can you, since you have the picture, the figure there, can you explain to me, so what you are doing is one in proportion of the other. So you have a control, so in this case was not urea, but nitrogen, nitrogen. And what you are doing is you are dividing the expression of one of the gene, for certain gene, in the stationary phase, and you divide it by the one in the urea. Is that correct? Or how are you getting only one column for this comparison? I would like to know how you are. Yeah.
This graph compare the result of the expression analysis using two different medium. So in the x-axis is nitrate condition.
So whatever is close to one shouldn't be significant, no? Because it means that the stationary and both control and urea are similar. Is that correct, Maria?
No, the condition is the same. It's still stationary and stationary, but it's just only the different medium.
Yeah.
Yeah, like in the x-axis nitrate, and this is the urea conditions.
Okay. Okay, good. So, do you think because at the end, many of those genes that you select that you already found in the transcript information, so you design, and you didn't know, you design your own primers, yeah?
Yeah.
And just to get an idea about the expression, and how the level was always in the same direction, and how it, because there's some analysis that allow you, when you have a gene network, allow you to plot that there. I think in your case could be interesting to, can you go back to that,
here?
No, the one with that you show us the different genes in the pathway, yeah. So, because what I think it's interesting to see if dependent how the feedback that they have, so if one is controlling the other, and if you expect the same, if the correlation that they have in the intensity and the magnitude, and there's some analysis that allow you to make some sort of, it's somehow to put that information about gene expression in a network, in a pathway of genes, and see if they are correlated and how they interact. It's more like a, so, and okay, your final recommendation will be, can you tell me about that, for if I'm interested in the culture of these, because what I want is to get that particular chemical, that could be used for anti-tumor treatment and so on. What is, with your result, what is your final conclusion?
The first is to extract the molecule of interest first. Yeah, and then we did the chemical extraction, and following that the chemical analysis, and then to do the do the bioassay to test them in the cultures in the cell cancers. Then whether it's to confirm is it kill the tumor cells or not. But then I mean all of this should be did in the urea medium, because my results is in shows high expression in the urea medium. So yeah.
And similar with marine question, this fold of change is similar to the other species that was reported, the fold of change in the expression. Yeah, for these genes that are involved in the pathway that generate that particular compound, is similar to those previously found in the other species that you mentioned.
The previous study was using transcriptome, like the different method. I use the real-time qPCR, but
but the intensity or the amount of could be measured somehow, no? Could be normalized and could be measured.
they only provide the general information of which genes are regulated and unregulated because of the porphyrin pathway is pretty big.
Yeah.
So yeah, they don't provide.
Okay, thanks a lot. Well done. Congratulations.
Thank you so much.
Thank you.
So thanks for this nice presentation. So my question is, I have some really basic questions. So when you started to look into the literature, okay, to find your gene of interest. So first of all, how much did you find? How many genes did you find? And based on what you selected your gene of interest?
Yes, I found 12 genes.
Because I mean, enzyme involved in synthesis of anti-cancer compound, and there is tons. So why make you choose this one specifically?
Okay.
I didn't get it.
So actually, in the model microbiology, all of the genes of or the enzyme are here. Even it's concluded that they are not in the green boxes. But when I check in the transcriptome of the Stylidroteca, it's only available. The genes that are available is only in the green boxes.
But in this specific diagram,
yeah.
So in the toolkit, the molecular toolkit they have tools. So does anybody look at the number of enzymes that can be interesting for your study, and or is it just the only one?
I think I'm the first person that study like the whole enzymes involved in the synthesis. Yeah.
Okay, right at the beginning, you were given this specific pathway to dig into and the enzyme. Okay. Then when you look into the transcriptome, so how did you process?
So in the transcriptome was like in the form of the Microsoft Excels. So I only type in the search, like there's a search section, like the name of the enzyme.
Okay.
Then if it's annotated in the transcriptome, like in the same name, like in the same enzyme. So after that,
okay.
Yeah, this is the
So
transcript.
it's based on the annotation on the genome. Okay, you selected this specific sequence, and then okay. So when you perform the blast, so how confident are you that the annotation of this genome is really accurate?
Yeah, I confirm with the blast.
Okay, and then what was the p-value of the
90%.
Okay, okay. Then when you are performing your experimentation with the area, so how long after the exposition to the area do you look at the transcript?
Within 1 month from the RNA extraction, you mean? From there?
When you perform your experiment with the urea, so how long after exposing the diatom to the urea, you are looking at the transcription inside? You are looking at your gene of interest. How long is the experiment?
To extract the RNA.
The RNA were extracted in the stationary and exponential. So the exponential within 2 weeks, and the stationary is within 2 weeks too.
Yeah, thank you. Okay. And did you never thought about doing a different temporal time?
Yeah, I didn't see that because usually the researchers only did in the stationary and exponential.
Yeah. Okay, thank you.
There is just time for a quick question. No one measured the chlorophyll production at the end. I mean.
Yeah, I didn't do that analysis. It
Because
should be nice.
sometimes in a metabolic pathway, yes, you have alteration in the enzyme production, but there is no change in the production of the final product of the metabolic pathway. I mean, could be. I mean, just to because at the end, the final interest is take this diatomer because it's useful in this condition to produce this compound. But you need to demonstrate that there is a significant increase in the production of that compound in that condition, not only by the genes that are altered, it's by the product.
Yeah. it will be nice in the future if we can do the like chlorophyll analysis and the extraction of the real metabolite. But yeah, this one only provide that the microalgae really have a big potential to be used as a future photosynthesizers.
And when you talk about genetic engineering, in what are you thinking? Genetically change the structure of the. What is your idea? When you said that these results are very useful for future genetic engineering procedures.
Okay, I have this annexes. So there is one research that conducted in the microalgae Cytotrium that the deletion of certain genes can lead a significant increase of the total lipids and the DHA production. So in this case, maybe we can like delete one of the genes of my microalgae to overexpress or suppress and to lead to enhance of the productions. Yeah.
That's it. Thank you so much.
Thank you.
Okay, last but not least, I know that. Are you ready?
Yeah.
Full of energy?
I am.
Take a breath and go ahead.
Okay.
Okay, the floor is yours.
So,
please.
Good afternoon, everyone. My name is Aina Lopez, and I'm going to present you my master's thesis called under the umbrella of reference genome, population genomics on the keystone species Paracentrotus davidus represented. And my supervisors were Carlos Carrera and Cinta Pagarolas. So to give you a little of context, now we're living in a biodiversity crisis that affects terrestrial and marine organisms. And from the different approaches to fight this crisis, genomics stands out because it allows us to obtain a high amount of data. These data are markers, and they're distributed along the genome, along the whole genetic information of a species. So they're genomic markers. And there are different types of genomic markers. They can be a sequence, they can be nucleotide, but all of them gave us informations about population structure, population connectivity. And genomics, to obtain them, usually relies on reduced representation sequencing or reduced representation techniques, because you can obtain those markers without the need of having the complete genome of your target species. You can have the reference genome, but it's not necessary. And when you work without this genome, you use the de novo approach because you do it from new. And this is really important for non-model species because there are those species that don't have this reference genome. At the same time, reduced representation sequencing or reduced representation techniques rely on restriction enzymes. However, when you work with this de novo approach without a reference genome, some considerations must be taken because you need to apply very restrictive filters, and even depending on the aim of your study, you still need a reference genome. So they remain an important asset in genomics. However, producing them is expensive, time-consuming, and you need large collaborative groups. Thankfully, there are projects and initiatives that ease this process of producing and sequencing, like the Catalan Biogenome Project or the Earth Biogenome Project, even in marine species, for example, the beluga whale, the Balearic shearwater, or the black sea urchin. So sea urchins are considered keystone species of the infralittoral benthic ecosystem. However, until 2018, only one reference genome had been published. So most of the studies on genomics on sea urchins use this de novo approach. The landscape is evolving, and in the recent years there have been publications of different reference genomes of sea urchins, including Paracentrotus davidus, the edible purple sea urchin, which is considered a keystone species of the Mediterranean and North Atlantic ecosystem and is threatened by fishing and rising temperatures. So studying the population status is important in order to understand if some measures need to be taken. Carreras et al. In 2020 did the population genomic studies on Paracentrotus davidus using the de novo approach because there was no reference genome available at that time. Now that it has been recently published, there is room for improvement, but there is also room in the so-called era of genomes, where a lot of genomes are being produced, to understand how important it is to have a reference genome on population genomics. Do you need to invest some of your money first on producing the reference genome and then doing the population genomic studies? And this is where this master's thesis plays an important role, because we aim to evaluate the importance of the target species reference genome in conservation genomics. Reduce representation techniques, but with the reference genome, versus the de novo approach. Which aspects do improve when you have this reference genome? So we address those questions through common use analysis in genomics, population structure and connectivity, genetic diversity indexes, mapping and distribution of genomic markers, and then functional analysis. So we use the raw sequencing reads from Carreras et al. Of 2020 that use genotyping by sequencing, a reduced representation technique. And the data were from 241 individuals distributed along 11 populations, 3 Eastern Mediterranean populations, Trieste, Fier, and Ryan, 4 Western Mediterranean populations, Culera, Xavia, Palos, Carboneras, 2 transition populations, La Herradura and Tarifa, and finally 2 Atlantic populations, Crozon and Sidi. So before showing you the pipelines, I want to make it clear that we obtained some genomic markers in this master thesis through using the reference genome published. So I'm going to call them reference genome markers. And we use another type of markers that were obtained by Carreras using the de novo approach, and I'm going to call them de novo markers. We use both in this master thesis, but the ones obtained by me were the reference genome markers. So here are the 3 main pipelines that we follow in this master thesis that were population structure and connectivity with the raw sequencing reads from Carreras. We map them to the reference genome, so we try to find a coincidence, and we genotype them, obtaining the reference genome markers. And with those markers, we can study the structure and the connectivity of our populations, and also see if some of them were significantly associated to environmental variables, because they're called candidate markers because they candidate to be under selective pressures. Then we have mapping and distribution of genomic markers. Using the de novo markers obtained by Carreras, again, we map them to the reference genome, and we try, it's like we have a puzzle and we put the piece on the genome, and then we study where they are located. If in exons, the part of the gene that translates into protein, introns, the part of the gene that doesn't translate into protein, or intergenic regions between genes. And similarly, we did this with also the reference genome markers. And finally, we go to the functional analysis, that using the reference genome markers, we see in which genes they are located. We obtain the transcripts from those genes, and we send them to an external server that will associate some labels to those transcripts. Those labels are called gene ontology terms, or GOL terms, and they give us biological process information about our transcripts. So we know if, for example, transcript 1 is associated with ion transporting, and then with the candidate markers, we can see if them are enriched in specific functions in comparison to the total markers. So the first results that I'm going to show you are the population structure and connectivity. With the reference genome, we obtain a higher number of genomic markers in comparison with the de novo approach. And when looking to the genetic diversity indexes that were the heterozygosity observed and expected and the inbreeding coefficient, the major discrepancy was this inbreeding coefficient, because with the de novo approach, they obtain a really high value, meaning that the reproduction was not random, but it was between organisms that were relatively close, while we obtain a more common value. So this high value was associated to a potential bias of using this approach, because as I told you, you need to apply very restrictive filters, but sometimes it's not enough. And this is considered a trade-off for these techniques, because you need to consider that you're working with a species without any kind of reference. So even when you apply restrictive filters, some errors can remain. Then the kinase analysis, what does is classifies your individuals into groups or clusters. And we obtain the same number of optimum clusters in both studies. So it was 2. So we had the Mediterranean and the Atlantic cluster. And this was the same Atlanto-Mediterranean structure in both studies because it was not cut clean. It was not Mediterranean and Atlantic, but more of a gradual transition over the Gibraltar Strait, because you can see that in both studies, individuals from Tarifa and La Herradura distributed to the Atlantic and to the Mediterranean. And this gradual transition has also been shown in other species too. And then, for a more internal structuring, we did the discriminant analysis of principal components. And in this case, both of them show a genetic differentiation over the longitudinal gradient, and it's a gradual between all the different populations. However, both dendrograms show 4 main clusters, 4 main groups that are the Atlantic, the transition, the Western Mediterranean, and the Eastern Mediterranean. And there is a high and significant correlation between both styles. So we obtain the same Mediterranean internal structuring with some minor changes. For example, with the reference genome, there is a higher differentiation between Atlantic populations. But overall, there is a really high correlation between both styles. So, our population genomics results mirror the one from Carreras. Same Atlanto-Mediterranean transition, same internal Mediterranean structuring, with the major discrepancy that was the imprinting coefficient associated to a trade-off of these techniques. So there is a big reliability of this de novo population genomics using the reduced representation techniques. Then we go to the mapping and distribution of the de novo markers, the ones obtained by Carreras. So mapping is trying to find a coincidence of your markers in the reference genome. It's like the puzzle I said. In our previous study, we used the reference genome, the closest available at that time, that was Strongylocentrotus purpuratus. And we only obtained 10% of our markers mapping. That means that only 10% could find a coincidence in the reference genome. Now, using the reference genome of our target species, Paracentrotus, we have a 90% of those markers mapping. So we have a big and significant improvement. You have a reference genome, you have more markers mapping. And this has also been shown in other organisms too. Then when trying to locate them, in which part of the genome they were, in both of them, most of them still in exonic regions, so the part of the gene that translates into protein. Then, and this shows an enrichment because, as you can see here, the genome composition of both the species, Paracentrotus and Strongylocentrotus, are not enriched in exonic regions. But what about the reference genome markers? Are they also enriched in these exonic regions? Well, from 6,000 that were in genic regions, 31,000 were in exons. So still, most of them map in exonic regions. Again, considering that the reference genome only had a 6% of exonic regions. So there is an enrichment independently if they are from the de novo approach or the reference genome. And this is because the restriction enzyme used. I told you that the reduced representation techniques rely on restriction enzymes. The one using this in Carreras was APEC1, whose restriction site contains 80% of guanine cytosine. The restriction site is the sequence that the restriction enzyme reads and goes and cuts, and you obtain your raw data. Considering that exons are enriched in exonic regions, there is a potential bias of this enzyme go on toward exonic regions. So the distribution of your markers is influenced by the restriction enzyme that you use. And finally, for the functional analysis, we did the redundancy analysis to obtain those markers that were significantly associated to environmental variables, and we obtained a higher number in comparison to the de novo approach. And now, when trying to see if some of them were enriched in some functions in comparison to the total, we couldn't, because from 2,000 total transcripts that we sent to the external server, only 300 were obtained, had associated Go terms. So we had really few transcripts with associated, and we couldn't see if there was enrichment or not. And sadly, this usually happens when you work with species that are very far away from humans or vertebrates. And as you can see here in this graph, that using the same software that we use, it tells you the annotated genes of each of the groups. And you see that most of them are on the 50%, so it's really low, but even some of them, it's low. And the only one that has more than 50% of their genes annotated is Craniata. So in this case, even when you have a well-annotated reference genome, the resolution is still limited because of the species that you are studying. So the big main conclusions of this master thesis were that reduced representation techniques provide reliable genomic data for non-model organisms even when you don't have a reference genome. That reference genomes improve genomic markers, characterization, and solve genotyping biases. And finally, that challenges remain with functional analysis and gene annotation, even when you have a reference genome. Considering that our results align with the ones from Carreras, there is not one genomic pool from Paracentrotus dividus, but different populations. so it's more vulnerable than previous result. So I want to thank everyone that was doing this master thesis with me.
Many people.
Yeah.
Thank you so much, Aino. Okay, go.
Yeah, I know. Great to see you here. So congrats for such an amazing work. So I don't know, it's 5:30 almost, and at least you keep my attention. So very interesting on the topic, but I think it's not an easy topic to present, and I really like the way that you simplify stuff to make it more accessible. So
thank you
for that. I think you need to really show the proficiency to do that. So that means that you know what you are talking about. So well done in that sense. So I have several questions, but of course I have to control myself and just use few questions. Then of course we have many people here evaluated to ask. So it's really, this is more philosophical thing. It's really de novo, or it's using a mapping with the different species.
It's the whole carreras is de novo, but the only thing that where they used the strongylation totus purpuratus genome was for their functional analysis.
Okay. So, it's not that they were mapped to a close related species. No. Okay.
No.
So that was clear to me now. Okay, great. So, because of course, definitely you are gaining. I mean, it's really clear. I think that you show it and you correct really well how that particular enzyme that you are using for digestion is enriching, even though the composition. That I think it's a clear explanation. You convinced me really well, so I really like it. So, but the thing is, at some point you have in terms of markers. Can you go back to the 3,000 versus 100, 9,000, something like that? Yeah, I remember. Okay, good. So, what about LD, liquid disequilibrium? When you are analyzing population, so are you considering for each breed that you have, are you considering only one marker? I mean, I know that you are working with small size, so very likely you don't have very small size of insert size fragment, so very likely you have only one stick per. Is that right, or you have more than?
No, we have one.
Okay, so, but on top of that, do you think that all of those are informative in terms of some of them could be fully linked and they are telling you more or less the same information, don't you? Depending the kind of analysis that you are doing.
Yeah, because I think that some of them are redundant.
Like, I mean, that's like every prune, depending the kind of analysis that you are trying to do.
Yeah, because, for example, here, the redundancy analysis in this case, the one with the de novo approach had explained more than the one that we use on the reference genome. So we have a higher number of markers here. We're working with 100,000, while in this case it was 3,000. So, and you still have less. So it's because some of them are redundant. So.
And in any case, and with that I promise I finish. So in your case, I think it's super nice. Apart from, I know that your aim, and you fully explained really well that your aim was to compare the reference versus no reference, and that's great. But in your case, I think I really, now since you put all the samples in the map, I would like to see some enrichment in the client, because at the end what you have is you have a client in salinity.
Yeah.
There's some software that allow you to say, okay, I'm going to see which of my genomic region, or more than one, associated with the client in certain parameters that you have. So to see, okay, which region along the genome with the reduction of the complexity of the genome that you have correlate with the change, so the change of frequency that correlate with what you might expect this pattern. Because in that way, even though you are not seeing geo enrichment, but you will see some region in the genome that could be somehow associated with that client in your environmental or whatever you do. Yeah? So I know it's not the focus of but there's some analysis that you can do on that. I think could be interesting.
There is no environmental data associated with the sampling. So I mean, you have environmental data associated with the sampling.
Yeah, we use I use the same one that we're using carreras that were through Copernicus.
I mean, on top of FST applies or whatever, there's something about clients that you can do. That could be
okay.
So thanks a lot. Well done.
Thank you.
Great job.
Thank you.
Okay, Laura.
Yes, thanks for this nice presentation, dynamic and very clear.
Thank you.
I'm not a specialist, but I'm curious about what what will come after your work. So my first question is, how did you select the markers that you studied?
Mm-hmm.
And
The 100
for instance, if you had more time, would you have got to others, or what is your opinion on that?
So the ones that we obtained, like the ones on the present study, we had to apply several filters to obtain them because we at the beginning we have 4 million of them. So we applied some quality filters and we work with them. But obviously, I think that now that Gonzalo also mentioned, some of them could be even more filtered. And the ones that are more interesting, that were the ones that the candidate, that are the ones that are under selective pressures, I think that this is where that we obtain a higher number because you can study more them, but even when the functional analysis, that we couldn't perform it, so it was kind of sad. Because we were aiming, we already did in my bachelor's, I already worked with this species, and we already did the functional analysis, and we obtained 0 results because we didn't have the reference genome. And we thought, okay, now that we have a reference genome, everything is solved. But it wasn't. And so now what we're, like the future perspective is to work with another sea urchin, that is Arbathelixula, and do exactly the same. But with Paracentotus, we're planning on doing whole genome sequencing. So to obtain even more data and trying to see if there are, like, for example, some differences, but just the reference genome that we use in this case is like just from one individual, and this individual is from the Mediterranean. So this can be a conflict with, for example, individuals that are from the Atlantic. So, but yeah, that's it.
Yes, your results show very clearly the different population.
Yeah.
Impacts, so
this one.
Yeah. So what do you think is the scale of the connectivity of those populations?
So
can you tell us what you know about that?
Yeah. So the thing is, over all, like the Atlantic and the Mediterranean are connected, especially through the Gibraltar Strait. But for example, the Eastern Mediterranean and the Atlantic, we found that in the individuals from the Eastern Mediterranean present some markers that were completely absent in the individuals from the Atlantic, and vice versa. So this shows us that there is genetic differentiation between both basins, but there is still connection through them. This connection could vary through the different oceanographic fronts that we have, and also because there is local adaptation, and there are different environmental variables in both basins. So yes, there is connectivity, but there's also genetic differentiation between both basins.
Okay. And if you consider the size of the basin or whatever, if you consider one generation of the sea urchin, what is the size in kilometer that you expect, like for population to have contact together?
That depends on the duration of the larval.
Yes.
Yeah, it's PLD or something like that, at least in Spanish. I think, if I'm not mistaken, that it was like 3 kilometers, but like they don't have like long PLD. So, yeah.
Okay.
Thank you.
Thanks a lot.
Thank you.
Well, congratulations.
Thank you.
Great work. Okay, so you give a lot of details. Okay, at the end, very compacted because, but it's also good because you wanted to explain everything, and it was also quite clear. Even for I am not a specialist in this kind of techniques, but I face similar problems because I also work with non-model species. We face with this kind of sequencing and so on the similar problems.
Yeah.
So the question is that this kind of approach, so how can be, or it is possible just to transfer to other species? So it's you see that it's an adaptation that could be done quite easily in other scenarios?
I would say yes, but obviously depending on the size of your, like the genome of your species.
Yeah.
Because like it's getting cheaper to obviously sequence a genome, but that doesn't mean that it's cheap because you need to sequence the genome, you need to ensemble the genome, you need to put all the genes, all the proteins, everything, and that's difficult, really difficult. And usually you work with the species with, or like even the grants, they give you for specific species that have very small genomes. So when you go to big vertebrates, it's even difficult. So I would say that depending on the size, it's feasible, but it will always depend on the species that you're working with.
Yeah, no, but so we work a lot also with other kind of invertebrates, or even fish.
Okay, yeah.
But of course, in fish, the genome could go through short genomes to very large genomes, and sometimes. So this is why when you were working in your markers with intron, exon, so it's very interesting because that makes sometimes difference in this aspect.
Yeah, when in my bachelor's that we did the same. Like, here, like we did the same, but not only with Paracentrotus, but with Arbacea, Symphodus tinca and Symphodus ocellatus, that are two fish species from the Mediterranean. But all, because none of them have the reference genome, we only obtained like 10% of those lossy mappings, so we couldn't see what was happening. We only could see that the restriction enzyme or the sea urchins enriched in exonic regions, and for the fish, it was going more towards intronic regions. But it was only 10% of our results because we couldn't study. So now the planning, I'm now building the genomes for those fishes, and the one from the other sea urchin is almost done, so it was published last week. So I will keep working on doing this with all the species that we work.
Thank you very much.
Welcome.
Thank you, Madam Ali.
Thanks a lot for this wonderful presentation. So I'm really impressed by the amount of work you performed during your internship. So when you were writing the lab, so the data were ready to analyze, or
Yeah, the data, because these were done in 2014 and 15, the recollection.
Yeah.
So when I arrived, it was the raw data.
Yeah.
So I have to filter it, mapping, and everything. But yeah, it was only the bioinformatic analysis.
Okay.
Well, only.
So you were talking about the functional analysis in your terms. So what is for you, in the context of your study, what are you defining with functional analysis?
Well, the functions that the genes that contain our markers in exonic regions have. Okay. So we could, this is functional analysis, but we really did, we tried to do the enrichment. So comparing what our candidates had some specific functions, if they were enriched, for example, in a function that is related with hypoxia or raising temperatures, in comparison to the total markers.
Okay.
Yeah.
Then this is all hypothesis. Okay. So if you have to change the terms, which term will you use?
The term?
If you have semantically, so if you have to change the term, what will you use instead of functional study or functional analysis?
Enrichment, enrichment biological analysis or something like that, because we only work with biological process information. We didn't count the cellular components or the molecular functions.
Yeah.
So it would say enrichment.
Potential
enrichment, or trying to do the potential enrichment and failing. But
because functional analysis clearly is when you remove a gene to look at the function, or you add a gene to look at the overexpression. So in the context of the work, I will not use
the function. Yeah.
It's just
Thank you.
Okay. Thank you so much.
Thank you.
Thank you. And you know that it's not. I am very surprised with your work and delighted with your work. But because when we were working with octopus, it was the same. I mean, using a SNP, it was not different for genetic differentiation. The pattern that we obtained, the previous one that we have with microsatellites, it was the same.
It was even with microsatellites.
Yeah, it was the same.
Okay.
But let me say something. When we are talking about the genomic studies, you have only one individual from some area. So are you expecting some changes in the patterns that you are showing us about genetic differentiation, structuring, etc.? If you sampling more individuals, because in some of these studies you are losing the intra-population variation.
Okay, yeah.
For example, in the case of Atlantic, you have 2 locations, one individual, one there. So
No, we have 20 something individuals in each population. Yeah, these are. I
did not see.
No, because it was I said here that were 241 individuals that we work with, and these are the numbers. Now I changed, but we have 20 per side, 22, 20 in the case of Dalian and 23 in the case of Crozon, but all of them.
That's the number of individuals. So 23 and 22.
Yeah, and 20 in one case.
Okay, I know that there are some people in the audience that want to make some questions, but I know I will be available, and she's already a master in science, so you can talk and take a coffee. There is a group picture now for teachers. Congratulations.
Everyone. Was that?